everyone, welcome back to cell biology class. In this video we're going to talk about facilitated diffusion. Facilitated diffusion is very similar to simple diffusion in that we are moving molecules down their concentration gradient. So we are moving them from where there is a lot on one side of the membrane across the membrane to allow them to come to an equilibrium. But a lot of molecules can't just go straight through that membrane. They may be too big, they may be too polar, they can't interact really well with that hydrophobic layer of the membrane. So they're going to come through a protein. So facilitated diffusion is protein mediated because these molecules can only move in and out of the cell if they have a transport protein helping them. In facilitated diffusion, these processes are going to be exergonic. That means that as a molecule goes down its concentration gradient, it's releasing energy. So the solid is going to diffuse in whatever direction its concentration gradient tells it to. If there's an accumulation of a lot of molecules outside of the cell compared to inside of the cell, that concentration gradient is going to tell that molecule to move in through the transport protein. If there's a big accumulation inside of the cell, those transport proteins are going to allow that molecule to move out. There's not any inherent directionality here, just whatever its concentration gradient says to do. If you want to look at this picture here, talking about the kinetics of it. So for simple diffusion, as you add more of your solute, more comes through the membrane. So you have an increase in the rate of diffusion. But for facilitated diffusion, at first, as you add more of your solute, you get a lot more of your molecule coming through, and even more than you get from simple diffusion, because it's really easy for that molecule to come in. But then you start to kind of slow down, and eventually you'll just level off. At that point, all of your transport proteins are letting things transport through them as quickly as they can. It just ends up basically causing a traffic jam. Adding a few lanes to the highway makes traffic go fast. But then just adding more and more cars isn't going to make it go faster. You're just creating a traffic jam. It's the same kind of thing happening here. As you keep increasing your solute concentration, they're driving through those tunnels as efficiently as they can. You're just going to have to wait for the cars in front of you to move. So in facilitated diffusion, because we are moving downhill, down that concentration gradient, no energy is needed. The influx or efflux of that molecule is all it takes. It releases its own energy. And facilitated diffusion can become saturated here at high concentrations because they're limited by the number of transport proteins available. Transport proteins are going to be big proteins because they've got to be able to have some sort of space inside to hold something. So they're going to be big. And they're going to be integral membrane proteins. You can see it goes straight through that whole membrane. It's integral, it's transmembrane. And they'll have multiple transmembrane segments. So they have to be able to create like each layer of this channel. Transport proteins can be divided into two different types. There are carrier proteins. These are carrier proteins. Sometimes they're called transporters, sometimes they're called permeases. And what these do is they bind molecules on one side, then they change their shape and release the molecule on the other side. So here, this carrier protein is open on this side, releasing it, but it can also switch open on this side, picks it up, opens, releases. Switches back, opens, picks it up, switches over, releases. That's how a carrier protein works. Channel proteins literally just make a channel. It's just a tunnel. Some of those tunnels are going to have specific limits to their size or to the charge of what they can carry. Some of those channel proteins are just anything that fits through and can go through. But again, both of these are going to be going down the concentration gradient. So you can see you are moving from where you have a lot of triangles to fewer, where you have a lot of circles to fewer. This could just as easily let things go out if you have a big accumulation inside the cell. Pores that you find in bacteria, mitochondria, and chloroplasts there are going to be channels here that are big, non-specific. Things that can go in and out can go in and out. They are usually made of a protein called porins. Here's a pretty little reconstruction of a porin. And they'll allow passages of solids up to about 600 angstroms molecular weight. Although these are pretty non-specific, most channels are going to be smaller and more specific. They're going to have stricter restrictions on what can pass through them. Ion channels are a type of channel that is involved in ion transport. And most ion channels are going to have specific requirements for what charge. So they let things with a 1 positive charge or an overall 2 positive charge or negative charge go through them, and also their size. Moving a solid through an ion channel is a lot faster than moving through a carrier protein because you don't have to do that conformation change. It's just constantly open. Things just flow in and out without having to wait for that protein to shift to release or pick up. Carrier proteins, on the other hand, alternate between open to the outside of the membrane and open to the inside of the membrane. Or in this image, open to the outside, open to the inside. So carrier proteins are an allosteric protein. They're allosteric. They have these two stereo shapes to them. They alternate between these two conformations. In this one state, the solid binding of the protein is accessible outside of the membrane. And in this other state, it just shifts right into that where it's accessible inside the membrane. And it just shifts back and forth between these. So if something gets picked up, it shifts and can go through to the other side. So you can kind of think of carrier proteins as being analogous to enzymes. There's a lot of traits that they have in similar to enzymes. One would be that facilitated diffusion involves binding a specific substrate to a specific solute binding site. So just like an enzyme has a specific substrate that it binds to its active site. The carrier protein and solute form an intermediate. So you have this, intermediate interaction of the solute and carrier protein, just like you get an enzyme substrate complex. Then after a conformational change, the product is released. And here the solute, although it's not modified, it's being released, just like in an enzyme. After creating the change, it will allow those end products to be released. And carrier proteins can be regulated by external factors. So if you think of some of the things that impact an enzyme, they are often likely going to impact some carrier proteins. So things like substrate concentration or inhibitors or temperature or pressure. These things can also impact how a carrier protein will work. Carrier proteins also have high specificity, which is also a trait we see in enzymes. They have high specificity for their substrates. A transport protein often is going to be very specific for one single compound. So here this one carries the orange circles. They wouldn't carry blue circles. They wouldn't carry orange squares. It only carries the orange circles. One example would be in erythrocytes, or our red blood cells. The carrier protein for glucose is specific to only a couple of types of monosaccharides. It wouldn't pick up a disaccharide. And it's even so specific that it is stereospecific. So only one isomer of that glucose is going to go through. The mirror image is not even going to be able to fit in. Another quality of proteins that's similar to enzymes is that they have saturation kinetics. That means that as you get more and more solutes, eventually you are going to just saturate your enzymes. So here, if you only have a little bit of orange molecules, you transport it through, and your transporter just waits until the next solute comes through. But if you had like millions of little molecules waiting, it'll just be transporting, then transport the next, and the next. So it has a maximum velocity that it can work at, and it just can't go faster, even if you keep piling on the work there. It is working as fast as it can. So that's what saturation kinetics is. Eventually, it's going to hit its limit, and that's as fast as it can work. There also can be competitive inhibition happening with our carrier proteins. So one example would be in this glucose transporter we were just mentioning. You could put another monosaccharide like mannose and galactose, and that could bind to your transporter and block it from being able to pick up glucose. There's a few different types of carrier proteins. Some carrier proteins only carry one specific solute, one single solute through that membrane. That's uniport. So here, this is molecule S is going through. This is a uniporter. Sometimes two molecules can transport at the same time. And if those two things go through that transport protein at the same time, this is coupled transport. Here they're both transporting through the same direction. They're similar, so it's symport. Sometimes this is called symport and sometimes co-transport, but I'll always call it symport. So those two molecules are moving through at the same direction. They're both moving down their concentration gradient. There's still no energetics being required to put into it. They're both moving down their concentration gradient. Sometimes you could have an antiporter, which allows antiport or countertransport. That anti tells you against. They're moving against each other's direction. So one is moving in and one is moving out. Both are still moving down their concentration gradient. So a lot of A outside is moving in and a lot of B inside is moving out. But they're moving in opposite directions through that one transport protein. So that is antiport. So in our erythrocytes, the glucose transporter is a uniport. It allows glucose in. Glucose comes down its concentration gradient. That's it. Then there's an anion exchange that is an antiport. So it will transport both chlorine ions and HCO3 ions at the same time in opposite directions. So this glucose transporter, this uniporter, it has these 12 transmembrane regions. So if you were to draw the whole thing out, it looks like that. And this is what allows the facilitated diffusion of glucose into the cell. And it goes through facilitated diffusion because there's a higher level of sugar in your blood. Your blood glucose level is higher than what you have inside of your erythrocyte. This transporter is called the GLUT, or specifically in erythrocytes, it's GLUT1. And if you folded this all up, rolled it into a little channel, you'd have something that looks like this. If you looked at it from the top, it looks like this. You can see here's a little channel where it just lets that sugar go straight through. So for this GLUT1 protein to allow transport in, first your transporter is open to the outside. That is a T1 conformation. And in this T1 conformation, glucose can come in and bind to that molecule. So now you've got your substrate bound, your solute bound. That causes this transport protein to change its conformation to where it is in the T2 conformation, where it is open to the inside. That allows glucose to be released. And then your empty receptor shifts back to the T1 conformation, where it's open on the outside and it can start all over again. Pick up another molecule of glucose. What's important to know here is that GLUT1 transport is reversible because this is facilitated diffusion. The only thing that decides which direction that sugar is moving is concentrations of sugar on either side of that membrane. So to make sure that we keep our glucose level low inside of cells, as soon as we bring it in, we phosphorylate it. So now we don't have glucose hanging around your cell. Now you have phosphorylated glucose, glucose 6-phosphate. So even though your glucose 6-phosphate concentration is now rising, it's harder for your cell to move that across the membrane. So that's pretty much going to keep your glucose inside of your cell. So once this is phosphorylated, it can't bind to that GLUT1 transporter. It's pretty much trapped in your cell. It doesn't have a way to get out. Okay, so as opposed to carrier proteins, channel proteins are more of just like open tunnels. They are going to be transmembrane channels. They're going to have a more hydrophilic environment in them so that your hydrophilic solutes can pass through it. And there's three main types of channels we're going to talk about: ion channels, porins, and aquaporins, which are kind of a subset of the other porins. Ion channels are tiny little pores because we're just moving one little ion through. And they're usually lined with hydrophilic atoms because those hydrophilic atoms are happy to interact with the charge on your ions. But they're usually pretty selective, not just for size, but also for charge. And when you combine size and charge, you end up with a pretty narrow group of ions that can go through. Most of these are just going to allow one specific ion to go through. So you can have sodium channels, you can have potassium ion channels, you can have calcium ion channels, you can have chlorine ion channels. That selectivity of the channel is going to be based on their binding sites, what amino acid side chains are there, and how big that opening is. A lot of ion channels are gated. That means there's some sort of gate that has to open and close. So there's a stimulus that opens the gate and then allows it to close back. One type of gatedness is voltage gated. So when there is a specific change in the membrane potential, that triggers this little gate here to either open or close, which allows sodium to exit or come in or be trapped. There are ligand-gated channels. A ligand-gated ion channel will be like this, where here you have a neurotransmitter binding. That neurotransmitter is the ligand. It is what binds to the receptor. And when that neurotransmitter or ligand is bound, the receptor is open, and so the sodium can go through. When the neurotransmitter is not there, here there's no little spots on that receptor, the ligand-gated channel is closed. It's got this little gate over it that the sodium can't go through. In a mechanosensitive channel, there's some sort of mechanical force on the membrane that forces that membrane to open, that forces that membrane channel to open. So here there's pressure on the cell membrane, which opens this channel. When there's not pressure on the cell membrane, that channel is closed. There's also some kinds of channels that are always open. Ion channels are really important in a lot of types of cellular communication. It's how your muscle cells communicate with each other to contract. It's how your nerve cells send signals from cell to cell so your brain can signal to your body what to do. Ion channels are important in maintaining salt balance in your cells. They're important in the airways and your lungs. One example would be the chloride ion channel, which is called a CFTR. CFTR stands for cystic fibrosis transmembrane conductance regulator. This will impact the ability of chlorine to go in and out of the cell. Having that chlorine means that it'll also attract water, which means that mucus gets rinsed off from that area. If the chlorine is not being properly transported, like in the disease cystic fibrosis, you'll end up with an accumulation of mucus, which has to be somehow removed to keep that patient able to breathe properly. There are pores on the outer membranes of bacteria, on your mitochondria, and on the chloroplast of a plant or an algae that are going to be larger, they're less specific than ion channels, and they're called porins. Here's a porin that you can see from the top view, how, pretty much anything that fits through that opening is going to fit through. And you can see here how their transmembrane units, they're kind of these flat arrows instead of these coils we had before. So these are beta barrels that form this because they're these flat beta folds instead of those alpha helices. And then aquaporins are a porin that exists specifically for water. They allow water to move quickly across the membrane. That movement of water across the membrane is a lot faster than what we would expect through just simple diffusion through the membrane. It wasn't until 1992 that aquaporins were discovered, which might seem really long ago if you're a college student, but that was within my lifetime. That was within my parents' lifetime, so this wasn't a very long ago discovery. So aquaporins are going to allow, here's like you can see these little water molecules go through. They allow water molecules to rapidly go through that membrane. That's important in your erythrocyte function. That's important in your kidney cells, which are trying to, detoxify these liquids. It's important in root cells in plants, which allow the water to be drawn up from the soil into the plant. It's important in those vacuolar membranes in plants where it holds water in the vacuoles. Aquaporins, though, remember, they are still non-specific. They're still facilitated diffusion. So water is still going to move in and out of the cell depending on where the solute is. It's going to seek an equilibrium here. Okay, we talked about a lot of stuff here in our facilitated diffusion. I'll see you back in our next video to talk about active transport. Bye.